Okay, good morning. We know they schedule us begin at 8 o'clock. This is also the fourth day for the convention. It's a little bit challenging. And so the chairperson, Stephen, asked me maybe to wake up the people, in the early morning, maybe try to give a more entertaining talk. Now, of course, I agree with that. But I'm under jet lag, so I will do my best, all right? As Stephen already told you about the title of this talk is the basics of using MRI for detecting cancer and pre-cancer cell. So since I have participated in this field a long time ago, almost about half century, so maybe my talk will be more focused oriented toward the historical development. So give you a historical perspective of how this MRI developed from one of the observer or a participant. Okay, now, well, it looks like that most of the people here, they are already experts on medical imaging, so I don't need to go through this slide. I was just thinking, because they APS advertised this as one of these UG-friendly sections, so therefore I prepare, to maybe to help the UG audience, but now it looks like not that many of them, so I can just go for very quick to skip. Of course, if we go to the hospital, there are major, I would say diagnostic technology could be good for imaging, would be like the CT scan, MRI. So one thing I think is very clear to this audience that they are very different because they use the X-ray to measure the density of tissue, and the MRI actually is imaging the physiological state of the cellular water. Now, this point actually become more important later, okay? Now, to the expert here, there's no problem, but to the audience, to the ordinary people in the society, it's very common to have a misunderstanding about MRI. So most people thought that MRI is an imaging technology of using magnetic field gradient to display the distribution of water proton in the body. But this so-called popular understanding is not exactly right. Since the water contents in most of biological tissue are very close together, so it's not possible to use the distribution of water to differentiate between the different tissues or between the different morphological state of the tissue, like between the normal cell and the tumor cell. So they have to use something else, okay? So this is just a nutshell of what MRI does, okay? So first it measures the nuclear spin signal from water proton. Of course, it's not limited to water, but water gives the biggest signal. And they actually use the MRI relaxation time to determine the physical state of the water molecule rather than the density of it. And then they use the magnetic field gradients to provide the positional information. And finally, you can use a fast Fourier transform or reverse Fourier transform to reconstruct the two-dimensional image. Now, so key requirement during our early development, the key question was that how to provide the contrast between the cellular water and the extracellular water. And also, we try to find out how to provide a contrast between the cancer cell and the normal cell. So this is the very early challenge. So how can the nuclear magnetic resonance produce a contrast in this image? This is really based on the difference in the relaxation time of the water proton. Now, again, even though this section is supposed to be UG-friendly, but now since there are not many UG there, so I can just go really just reminding the audience that these nuclear relaxation, the nuclear spin relaxation time we're talking about is the T1 spin relaxation time and the T2 spin relaxation time. So in a way, they just measure how the system reaches a thermal equilibrium for this nuclear spin system. So we know that, the nuclear spin will just possess around the magnetic field, the local magnetic field in the river Lama frequency. So if you look at the ensemble of the system, so at thermal equilibrium, then the nuclear spin actually will give you a net magnetic moment along the z-axis, which is the direction of the magnetic field, and there's no nuclear magnetization in the transverse plane, in the XY plane. So if you disturb the system, then how quickly can you build up this longitudinal nuclear magnetization is characterized by the T1. And if you disturb the system, and then how quickly the transverse magnetizations kind of go away and depend on the T2. And that from the name you can see the T1 is mainly due to the interaction between the spin system and the structure of the system, and the T2 is mainly due to the spin-spin reaction. Now, so very early in 1950, so physicists already find ways to develop this spin echo MMR method to measure this relaxation time. For example, here you can see using a pulse 1, this is a like 90-degree pulse. You will tip this nuclear magnetization from the longitudinal way to the transverse plane. And then sometime later, you have another pulse called a 180-degree pulse. It will refocus this nuclear spin, and then it will have an echo later. Now, actually, you can use a cartoon to show that. But again, there are not many students over here, so I can just skip the time and not to worry about it, okay? Now, so let's go back to the major question, particularly in the early days of the developing MMR imaging. And that is, how can one detect the cancer and the pre-cancer cell using MMR? Now, I put both of them over here because just detecting cancer is not enough. You know, as a useful diagnostic technology, you also need to detect the before the tumor is formed, before the tissue becomes cancer. Okay, because that gives you an early treatment. You can have an early medical intervention. Okay, that will help the patient so not to go through this cancer development. Okay, I was fortunate to be involved in this in a very early day. Actually, when I was a PhD student at Rice University, we built a homemade spin echo MMR spectrometer to study the transport properties of helium-3 and helium-3, helium-4 mixture. It's a superfluid. So I was working in low temperature physics, you know, just studying quantum fluid. But I started to develop a very strong interest in biomedical area. So biophysics, biomedical physics, I think that's the future to go for me, okay? So I took a lot of course, actually, or sitting some of the course in my graduate year. So when I finished my PhD in 1970, so I collaborate with a young physiologist called Carlton Hazelwood at Baylor College of Medicine. And we decided to use the spin echo MMR method to study the physical chemical state of the cellular water, Okay, so at that time, the water sample is taken from the rat, the skeletal muscle. This is called gastrocnemius. We take this sample and then put in our home-built MMR spectrometer to measure the relaxation time like T1 and T2. So the first thing we found is that the relaxation time of water proton in biological tissue were very different from those of the ordinary water. And the second thing, which is more interesting, is that the relaxation time of the water molecule changed during the tissue development. Now, for example, here, let's compare three groups. This is the pure water. This is the mature muscle. You can see the mature muscle has a shorter relaxation than the pure water. But the immature muscle has a lengthening relaxation time compared to the mature muscle. So that means both the T1 and T2 in the muscle cells, it becomes shortened from the immature state to the mature state. So what this tells us is that the physical environment of this cellular water will change during the normal animal development. Now, this has a strong implication because we know tumor development or cancer is known to be associated with dedifferentiation. So it goes the opposite way. Now, if the relaxation time shown in MMR is depending on the developmental stage of the biological tissue, then this will give a possibility that one can using, by measuring the MMR relaxation time, you can determine whether the cell is in a normal state or the cell is actually de-differentiated and go to the tumor state. So this has a very strong indication. So that stimulates us to start to use the MMR to study tissue water in between the cancer cell and the normal cell. But at the same time, actually, we have a competitor, Dr. Raymond Domanian from the Downstate Medical Centers. He actually is a friend of my collaborator, Carlton Hazelwood. Okay, and he also had the same idea as we have, right? And so he quickly used a commercial equipment to make some preliminary study of a three type of tumor here. The Walker sarcoma is a muscle tumor, and then also the hepatoma is the tumor in the liver, and also another tumor. He found at least in two of the tumor, if you compare the T1 in the tumor cell to the normal cell, like compare sarcoma T1 with the normal muscle T1, there is a significant link in it. And the same thing also with the hepatoma. Now, the data is relatively small, but this is new. Okay, the important thing that why he was able to publish in Science is because, actually, according to most people, this is maybe the first publication referred to the use of the new MMR relaxation time to differentiate. Now, at the same time, of course, we are also doing work based on our earlier finding. Well, in fact, the Domanian also acknowledged, this work was partially inspired from our work too. Okay, but we use a different system. Instead of using multiple tissue, we use a single tissue, and this single tissue is the mammary gland. Okay, now because why we do that? The reason is the breast cancer at that time in the 1970s is known to be the #1 killer for women. Okay, so if you look at the women dying of cancer, the most frequent case is the breast cancer. Okay, now I haven't really followed about the cancer study. I suspect even these days, breast cancer is still a major killer, one of the major killers. #2, okay, well, good for helping out. For women. So we choose this system to study because it has a very strong clinical relevance. Now, there's a second advantage is that we collaborate with a pathologist, and so we develop. So he developed a mouse model for this breast cancer. So we have tissue, a three well-defined morphological state. One, of course, is the so-called normal mammary gland. And the second one is the pre-neoplastic nodule. So this is a nodule, you can feel it, but it's not quite a tumor yet. And finally, you have this neoplastic tissue, which is the cancer, the breast cancer. So we have the same kind of tissue, but at three different, well-defined, distinct morphological states. Now, our study also not only studied T1, we also studied T1, T2, and the spin diffusion coefficient. So in every category of this physical measurement, there was a clear statistical difference between the relaxation time of the tissue water at different morphological states. In fact, this work was reported in the same meeting as today, at the APS March Meeting. But this time is since, it's go back to about half a century, okay, more than 50 years ago. Well, you can guess how old I am, okay? I mean, unfortunately, I can still stand up here and talk to you, and I see it as fortunate, right? So in fact, that our study, we reported at the 1972 APS March Meeting, which was held in the Atlantic City in New Jersey. And so after the meeting, and the APS officer sent me this a news clip, and they say this is their press release. The title is very impressive. They say nuclear physics aiding in breast cancer detection. Now, this is a very attractive title. Well, actually, it took me some time to realize why they want to do that, because we are not the big guys, but why they picked our study as the feature report. There was a reason in it. And the reason is nuclear physics has an image problem. Now, recently there's a movie called Oppenheimer, which talk about the Manhattan Project, the building the nuclear bomb. Oh, by the way, how many of you have seen Oppenheimer? Well, only half of people. It's disappointing. I thought the APS people in the APS meeting, maybe 90% of the people have seen that movie, but that's not true. Okay. Well, anyway, but it's a good movie. I think they're going to get some big prize for it. Okay. And from that, you can see most of what they talk about is how they use nuclear physics to kill people. Okay, to develop the weapon. Now, this is not good for the image of APS. So APS want to give some example that nuclear physics not only can kill people, you can use nuclear physics to save people, to save life. Then, of course, this nuclear physics refer to MMR. MMR is not exactly the same nuclear physics in the April meeting, okay? But anyway, it helped, okay? So you can see that, so people are very excited. And also, this tell you, this is a novel idea. It's a novel idea. How to use the MMR, to save life for medical use. So I would say the medical physics go a long way. And this is just one example to tell people the physics can be very useful in medicine or in helping people. And 1 year after we report our finding in the APS meeting, and Professor Lauterbach published a short paper in Nature in 1973. Now, this paper is not about using MMR to detect cancer. It has nothing to do with it. This is just an idea, actually a very straightforward, a simple idea on how to make imaging using the magnetic field gradient. Okay, so therefore, even in his title, he said that this is example employing nuclear magnetic resonance. So he's not claiming using MMR for medical use and that sort of thing. And because of that, so he did not sign the paper with the median. He did not sign our paper, okay? But soon after that, Professor Lauterbach and Dr. Deranian started getting a big fight. The reason is because both men, so both scientists, are more than ordinary scientists. They are entrepreneurs, okay? They were very enterprising scientists. Deranian, soon after he published the paper, he wrote a patent, he applied a patent for a device based on MMR to detect cancer. And Dr. Lauterbach is not only a chemistry professor at in the state of State University of New York, he also is the executive in a small company called MMR Specialty. Okay, and he in the early 70s, he also tried to apply a patent for this magnetic imaging. He called it the chromatography. Okay, but I saw some book describing that, and his patent application was not successful because the lawyer think it's difficult to make that claim. Okay, so they dropped the patent application. But still after that, so he is a very strong promoter, and the same as the Deranian. So both of them are very active promoter of using MMR as an imaging device. So actually, they are dominating field because they come out to all the meetings and say, well, I invented this MMR imaging technique. And the other guy say, I invented MMR imaging technique. But the situation was getting worse as when in 2003 that the Nobel Prize in medicine was given to Lauterbach, but not to Deranian. So Deranian was very upset. Now he built a company called Fauna, and in the web page you can still see it. I think today that he thinks that this it was shameful that Nobel Prize excluded him. Okay, and in fact he placed a full-page ad in New York Times, and this is the title of the ad. He said, "Well, this is the proof that the shameful wrong must be right," and then he go on with a lot more detail, saying that why he is the one who deserved being the inventor of the MRI imaging for medical use and not the other. Okay, now when I say, well, I just try to be entertaining, all right? Because personality does come to play a role in science development, okay? So why the maiden was so upset? Because in his view, okay, people look at things in different view, okay? In his view, he thinks it's illegitimate. So he thought, well, what does MRI measure? Well, it does not measure really the density scanning. It's really, in his way, he said mainly T1 scanning, but later actually we found T2 scanning is more useful, but actually you have a mixing of all this. Now, if you look at, what are the key development for the MRI imaging? So the first is the discovery of using MRI relaxation time to differentiate the relaxation time difference to detect the cancer. That's the first development. The second one is use the magnetic field gradient for scanning. And in his mind, he thought Rutherford only contributed to number 2, but not number 1. And he, on the other hand, contributed to both number 1 and number 2. And so therefore, that's how he feels. And then he feels that the Nobel Prize in medicine did not recognize that. And so he was really upset. Now, competing for credit is not something new. This is it's quite happen all the time in science. This is somewhat an extreme case. But I think it's no use to second guess the decision of the Nobel Committee. Okay, and sometimes getting a big prize also depends on one's luck. Okay, so be it. So therefore, we are not going to really talk too much about it. So instead, this is a science meeting. Let's just focus on the scientific question. During the early development of MMR, actually not, I would say not only early development, even today, some of these questions are still relevant, all right? So what are these key questions? Okay, first is how reliable is the MMR measurement in differentiating cancer cell from non-cancer cell? The second question is, can the MMR image provide sufficient resolution for clinical use? And then the third question is, what is the physical mechanism underlying relaxation time change between the normal cell and the cancer cell? Now let's look at question one by one. Okay, let's look at the question number 1. How reliable is the MMR measurement in differentiating the cancer cell from the non-cancer cell? Well, this is a very relevant question. And that is because in the early 70 and 71, after the Maiden published that short science paper, many people questioned it. Particularly, I remember a scientist from Johns Hopkins University, Donald Hollis, has wrote several papers, later even wrote a book, that saying that the T1 lengthening could be caused by multiple, maybe other, so other things other than cancer development can contribute to T1 the lengthening. So T1 lengthening alone is not sufficient to say that this is the cancer cell or not. Now, in our case, we do more than the median, of course. Okay, so we have, in fact, as I told you, that when we look at these mammary cancer animal model, we have a very clearly defined 3 different morphological states between the normal mammary gland and the pre-neoplastic and the neoplastic tissue. And you can see the statistic here is very, very clear. So one can clearly differentiate between one morphological state to the other morphological state. So the statistic is very clear there. But this is the animal model state, okay? To be clinical relevant, you have to study human sample, okay? And so we did that. Actually, a few years later, we collaborated with the pathology in the Texas Medical Centers, and we looked at this biopsy sample from human and also from the breast cancer. Okay, so again, so we published this in the Journal of the National Cancer Institute, and showed that nuclear magnetic resonance study on the human the breast dysplasias and neoplasm. And the conclusion of the paper is that our results indicate that MMR relaxation time could distinguish between the breast neoplasm and other disease with a p-value less than 0.001. And given a single sample, the probability of classifying it non-neoplastic or carcinoma could be accomplished with an 85% confidence. Now, this is quite significant for pathology. And then for human breast tissues, we think the relaxation time T2 may be more discriminating than the T1. Okay, and so that's what. And so now, you can design a MMR machine that will monitor not only just one single parameter, like T1. Okay, and you can have a multiple MMR parameter. Now, what about in the second question is that so can the MRI image provide sufficient resolution for clinical use? Well, if you look at the early time, in the 1970s, it's far from that. So this is the just examples contributed by those three groups. This is by Rodenberg on a mouse. This is by the fauna image produced by the Median group on a human chest. And this is an image produced by Mansfield's group. Okay, and you can see now these MMR images were based on this spin density measurement. It's very little use, because there's no good anatomical detail to allow you to make any identification. But starting from 1980, things changed because now they have the new generation of machine. They based the MRI image on the relaxation time measurement. And you can see that this is much more clear, and you can see the general feature of these anatomical structures in the human body. Now, this, of course, is not the limit of that. And then later, and also this is starting in 1980, but it's getting more popular. It's called the spin warp MRI imaging, which will use a combination of pulse, the RF transmitting pulse, the receiver pulse, and also multiple magnetic field gradient, not only in one axis, like in the z-axis, but also in x-axis, y-axis. And you turn on, turn off this magnetic field gradient in a pulse form. By doing this careful combination of this different kind of manipulation, and you can come up with something far more clear. For example, you can see this is just an example showing that the spherical pulse sequence used in the spin warp MRI imaging is like this is the RF in the transmission pulse. This is a slice frequency phase are different types of magnetic field gradient pulse. And finally, you get the signal. From the signal, you can now go for the reverse Fourier transform, and you can generate a two-dimensional image. And if you do it in multiple slides, then you can generate a three-dimensional image. And that's what that people are doing today. Actually, Michael here has a very advanced MR center, and too bad I can't listen. But this to you guys are now become routine, okay? And so this is what people kind of attempt with this. I would say more advanced MRI technique. And you can see that yes, clearly there's sufficient information, sufficient detail to allow the physician to make judgment whether a cancer is developed in one patient or not. Okay. Another improvement is the use of the contrast enhancing agents such as the gadolinium, and this is one example, and this is on the breast tissue. Before you apply this gadolinium agents, you don't see the tumor, but now after you apply this contrast agent, you can see it very, very clear. Okay, so now I see my time is almost up, so I have to really rush now. Okay, So about the first question, so what is the physical mechanism underlying relaxation time change between the normal cell and the cancer cell? Well, this is a complicated question. I would say the research is still actively going to that. Okay, now you see, if you go back to the 70s, in the early time, the biologists are debating what is the physical chemical structure inside a cell. You know, one of these, I would say, popular view, which is shown in most textbooks, is that the cell is just a set of biochemical solution enclosed by membrane. But starting in about 1980, people changed their mind. Actually, you can see nowadays, they're using different types of microscopy. You can see the cell is a highly structured system of iron water macromolecular complex. So these complex, for example, I also in the 80s, I done some work with Tom Lee's NIH using this phase etching, deep etching, rapid freezing, phase spectral electron microscopy. You can see, and this is the work produced by another science Asia will cover. You see, this bar, this gap bar is a 15 nanometer. So you can see the water, the cellular water, is never too far from the surface of the macromolecule. So they're always within about maybe 10 nanometer distance. So the water inside the cell, the majorities are interfacial water. They're not bulk water. Okay. Now for that, you can tell, you know, even if the artificial physical system, like between two pieces of glass plate, or between this membrane layer, you trap the water there. You know the water trapped over there in the distance of nanometer will have a structure quite different from the ordinary water. And besides that, there's also active exchange, you know, between different pools of water. Some of those are fast exchange, some are slow exchange. So the whole situation is very complicated. Okay. Now, in fact, because we know the mature cell usually is in the interphase state in the cell cycle. And the tumor cell or cancer cell, most of them in the mitotic phase. And there is a difference in the structure, the macromolecular structure in this between the interphase cell and the mitotic cell. And this structural difference can also affect the physical chemical property of the water to it. And in fact, so my time is up. So we did that study. So now this is my slide, conclusion slide showing the contrast of MRI image does not come from the difference in water content. Instead, the MRI contrast comes from the difference in water relaxation time. And the change in relaxation time reflects the variation in the physical chemical environment of the tissue water, and the degree of this ordering in the cell water allow one to determine whether the tissue is becoming cancer or not. Okay, so thank you. Thank you.
Okay, we have time for maybe one question.
Thanks for all that great history. The T1s in the breast that you showed that they're good, malignant versus benign, they're clearly different. But when you do clinical breast MRI, you have to use a contrast agent. Nobody would ever trust just the T1 differences, and that would be kind of, that's disappointing. Do you know, are we just not doing it well enough? Could we, you know, magnify somehow that T1 difference that you saw? Because seems like we sure need gadolinium to be able to see breast cancer with MRI.
You know, I have literally my hearing is not terrible, but I get the general sense. So what's the key point in the question?
Oh, I think he wants to know, do we have to measure T1 and T2 better because we don't use it really to differentiate? We use gadolinium.
Yes, actually what I referred to this was in the early days. So we can design the parameter, just target only to one kind of relaxation time, T1 or T2 or the spin difference coefficient. But now that using the imaging, because they're using so complex of these post program, it's very difficult to have a clean study. Just you know, using T1 as the imaging, T2 the imaging. So usually they come as a mix because that's, you know, this is the time to play because if you want to design your whole system to give you the best anatomical structure, but then you cannot have a very pure physical state to go with. Okay, but so now actually it's a combination of T1, T2. But in our early study, when we do this isolated study, in our opinion, we think that T2 is more telling than the T1.
Thank you.
All right, let's move on.